What is you put show navigator?
That just seems. I've never presented from this laptop before. But I can't.
Actually, it does have the British time.
Yeah, no, I know it's not very helpful. In fact, let's take that off. But I will put on the time.
And with right click, right on top of.
No.
Spoiling.
It's alright, doesn't matter.
Okay. Okay. Yeah, no, I don't know how to do that.
No, that's fine. That's fine.
And maybe.
The best one.
When this happens, my wife said that how many bedrooms?
How many?
¿Cuántos hombres se necesitan para hacer? Cambió de.
Porque es PowerPoint, ¿no? No, Keynote. Ah, y el Keynote tiene. Keynote have that printed? Sí, hay que volver a donde está esto, a donde está la presentación. Ah, pero hay que cambiar el. Ah, yeah.
Except I can't see them.
Side to X.
Where's the mouse gone? Let's try that again. Right. So they're there. You see, you can't see the mouse.
No, it's the opposite. It's changed.
So I can't see the mouse anyway. And there must be a way of changing it on here. View. Can't see it on there.
What about here in view? If you go there in view.
There.
No, this one.
There.
And slide only? No?
That'll just show this, I think. We can try, but yeah, so that's just showing, yeah.
Sí.
Right, so it's just that you can't see the cursor.
Where is it? Disconnected.
Maybe this field is at the right.
Did you see it?
Yeah.
Oh.
Es que le cambió una random formato y lo que él quiere mostrar en la pantalla está en su computador y se va a ver. No, pero es con Keynote más todo en Mac es horrible.
Maybe you need to reconnect the.
If I close it and I open it again, it won't. Maybe it won't have saved the. Let's try that.
Yeah.
Yeah, we're trying to work out how. Let's see. It's switched to.
¿Y se puede presentar un puntero? ¿Tiene puntero? Ahí está. Show the pointer. Yeah.
So maybe now.
Ahí.
There you are. There we go.
Cámbiala. Ahí. está de puntero.
Thank you very much.
Okay, so I'm going to introduce you to the dean will say something words and then okay, ¿comenzamos? Hola, buenas tardes. Bueno, gracias por asistir a esta actividad importante para nuestra facultad, pero bueno, principalmente veo gente de microbiología porque es un tema que está muy relacionado con el departamento, así que es muy bueno verlo aquí. Les voy a presentar al profesor Benjamín, probablemente ustedes ya lo han visto en redes sociales. En Spanish es Benjamín. Él, bueno, es profesor de la Universidad de Anglia en Inglaterra y nos va a mostrar un poco dónde está su campus. So I'm going to switch now to English. So I'm going to introduce you to Dr. Benjamín from the University of East Anglia. He's lecturer in the Norwich Medical School, if I'm correct. And well, I have the pleasure to have you here in Concepcion. We did our PhD in the same laboratory, so we are part of the same root coming from that in some way. And before your talk, and obviously thank you very much for your visit here in Concepcion. Our dean will say some words to the audience before starting with your talk. So thank you very much again, Ben, for being here.
Thank you.
Thank you, Dr. Evan, for this talk. Bienvenidos a todos, buenas tardes. Les queremos dar la bienvenida y el agradecimiento que participen en esta clase inaugural del posgrado que gentilmente nos ha brindado el apoyo el profesor Andrés para poder desarrollarla. Esperamos que sea de todo su interés y pueda ser provechosa para ustedes. Hoy día el tema de la resistencia bacterial a fármacos es un gran tema, lo conversábamos esta mañana con el doctor Graf, pero también plantea los desafíos de la interdisciplina y el trabajo colaborativo con otras áreas del quehacer de la microbiología. Por lo tanto, creo que también aquí va a haber una apuesta, un desafío, a que ustedes puedan de alguna manera, ¿cierto?, trazar esos caminos, esos puentes hacia la interdisciplina, hacia el contacto con los estudiantes de posgrado de los otros programas de magíster y de doctorado que tiene la facultad y poder enriquecer su propio trabajo a partir de estas experiencias y de las oportunidades que aquí se generan. Así que, doctor Opaso, muchas gracias a los dos programas por apoyar esta actividad del posgrado de la facultad. Dr. Evan, I would like to say thank you again for staying here and enjoy your presentation.
Thank you very much.
So thank you, Nate. Before just starting, I forgot it, but now I just remember in time. So also we would like to thank the UK Embassy in Chile because they supported his visit and they were very keen to all the organization of that. So thanks to them, Dr. Evans is here with us. So thank you. And this presentation also is being transmitted through YouTube from our channel of the faculty. So at some point, if you would like to watch it again, will be available there for whatever you want. So thank you again, Ben, for being here, and we are now very excited to see your talk. Please.
Thank you very much. Thank you for that welcome. It's a pleasure to be here. It's great to have been invited. This is my first time to South America entirely, so it's been a fantastic visit. And I'm very excited to show you some of the kind of work that we do, where I'm from in Norwich. But first of all, I'm just going to tell you a bit about where I am from. So I'm from a city called Norwich in the UK. So that is over here, you can see. So we're a little bit out the way. We're near the coast. We're in the countryside. We're a small city, about 150,000 people. So it's pretty small. Very old city as well, founded in about 43 AD. We have a castle and a cathedral, lots of really nice old picturesque looking streets. And we're also a UNESCO World City of Literature. It's one of the things that Norwich is famous for. So it's a beautiful place to come and visit. I'd very much encourage you all to come here. It's a really nice place. But we're also known for our science, and that is because of the Norwich Research Park where I'm based. And this is what it looks like. I think this is a drone shot that's been taken with a fisheye lens to show it all in. But we have the university and another of the other research institutes on the same site, which makes up the research park. And I wanted to highlight this to show that whatever area of microbiology you might be interested in, we'll have someone who does it. So we're very keen on collaboration, and if you are interested and want to get in touch, we'd love to hear from you. So we have, as you can see there on the right-hand side, we've got the hospital. So this is the main hospital for the region, large university teaching hospital. Over in the distance there, we have the university main campus, UEA, but we have, these are the playing fields in between. This is also a university building here, and then this is the building I am in over here. So the university buildings are spread right across this campus. It's about 1.5 miles long. Sorry, I don't know what that is in kilometers. But a reasonably big campus. And then we have these other research institutes. 3 of these are UK government funded institutes. I think there are 7 in the UK altogether, and we have 3 of them on the research park, so it was quite important for us. The first of these is the John Innes Centre. They do plant science, genetics and microbiology, so the particular interest in things like looking for novel antimicrobial compounds from soil organisms, as well as plant diseases, microbial diseases of plants, and the interaction with plant roots, these kind of questions. We have the Earlham Institute down here. So this is one of the UK centres for genomic analysis. They are tasked with coming up with new approaches for genomics. They're also one of the main centres in the UK for sequencing whole lot of genomes of various organisms so that we can catalogue all of life at the genomic level. And over here we have the Quadram Institute. Here they focus on food science and gut health and microbiology, so the link all the way through food microbiology and the gut and your health, linking all those aspects together. And then we have a privately funded research institute here, the Sainsbury Laboratory, and they do similar things to the John Innes Centre. It's plant microbial interactions that they're interested in, molecular interactions, to help secure our food chains, develop new plants and combat disease. And at the university itself, in terms of topics related to microbiology, we have medical microbiology, which is the area I'm in. We have a lot of environmental microbiology, and we have a very strong area in climate research as well. And climate obviously impacts everything we do in a microbial sense and a disease sense. So we're one of the largest clusters of microbiologists in the world. We have over 100 microbiology research groups on the research park. And we formed this Centre for Microbial Interaction. So it's not a physical centre, a physical building. It's an umbrella organisation that brings together everyone on the research park to do with microbiology. And we're here really to promote these areas of research into food security and health and disease. And the main mission we have is in driving excellence in these areas and in reaching out and collaborating with people from both nationally and internationally. And that's why I'm so delighted to be here speaking to you today and to make these connections. If you're interested, that is the web address down on the right-hand side there. And if you go there, you'll see our 6 main themes in the centre. And you can see that these cover all aspects of microbiology. I'm not going to go into them in detail now. You're very welcome to go and have a look. But any area that you're interested in, we will have researchers across the research park who are potential collaborators for you. So do go and visit us, have a look at the website and see if there are your future collaborators are there. Okay, so moving a little bit more onto me now, where I've come from and where I am now. So as Andres said, we met in Edinburgh doing our PhDs together, and there I worked on an organism called Acinetobacter baumannii, which many of you will be familiar with. Being the one I did my PhD on, that's my favourite organism, so a lot of what I do is now focused on that area. I then moved to the University of Manchester and did a postdoc there with Streptococcus pneumoniae, and there we were looking at homologous recombination. I then did another postdoc at University of Liverpool, Pseudomonas aeruginosa in cystic fibrosis patients. And then I got the opportunity of a lectureship at Anglia Ruskin University, that's ARU there, on their Cambridge campus. That's a teaching university, so I spent most of my time teaching, but managed to do a bit of research on the side there, and then finally moved to where I am now at the University of East Anglia. And in both of those positions, what I'm focusing on is drug resistance and pathogen adaptation and evolution. And you can see from the Venn diagram there, I've kind of moved between these different disciplines, but I now see myself as sitting fairly in the middle of these kind of areas. I'm not an expert in ecology or evolution. I'll go and speak to the experts and them and bring them into what I'm doing, but I think what I do spans these areas. So what I'm planning to speak to you about today, these are the three main research areas that I have at the moment. You can be summarized really as antibiotic resistance in gram-negative bacterial pathogens mainly, a little bit of gram-positive, but it's mainly gram-negative pathogens. Non-antibiotic drugs as drivers for antibiotic resistance evolution, and the role of the chromosome structure on drug resistance. So the first of those topics, antibiotic resistance in gram-negative pathogens, this is the kind of research that I know a lot of you guys are actively engaged in now. I've had the pleasure of hearing about some of the students' work here while I've been here, the fantastic work you're doing in this area. And we do similar work. These are just some examples of some recent publications. And I love phylogenies, so almost all of my papers will have a phylogeny in if I can get one in there. And that's the type of work we do, similar to some of the studies you guys do, using genomics and phylogenetics and molecular biology to look at pathogen epidemiology, the resistance mechanisms and the potential evolution of those. So that's an area of interest, but it's not what I'm going to focus on today. I'm going to focus on the other two areas because I think they're probably areas that people think about less, and so I want to focus on a bit more. And the first of those is the non-antibiotic drugs as drivers of resistance. So what am I talking about there? Well, there are huge numbers of types of drugs that we use for all kinds of different purposes. This paper here, they took a number of different drugs. These are the different classes of drugs shown down here. The number here is the number of drugs in that class that they tested. And they tested them against a number of commensal organisms, human commensal organisms. And then the number here is the number of those drugs that had activity against the commensal organisms. And you can see that in terms of the color scale, that's the number of affected strains that were found. So as we'd expect, antiseptics, antibiotics, lots of activity against commensal organisms, no surprise. But you start going down the list and you see things, well, antiprotozoals, antifungals, antiparasitics, antivirals, and then human-targeted drugs. So these aren't even, these are not supposed to be targeting bacteria, but they still have antibacterial effect. These aren't supposed to be anything to do with infection whatsoever. So these are other drugs we use in healthcare for various different purposes, but they have antibacterial properties. So the question is, should we be worried about that? We know bacteria respond to selective pressures very easily. You give them antibiotics and we get resistance emergence. So what happens if you give them these drugs? What does that do to them? And is that linked to antibiotic resistance? So one of the areas that we've focused on is cancer chemotherapy drugs. Lots of these drugs were originally discovered many, many decades ago because of their antibacterial effects, but then they were repurposed for use in cancer chemotherapy. Antibiotics were seen as being much better. So it's not new information that these are antibacterial, but we want to know how much do these affect bacteria? Can the bacteria affect the chemotherapy? And can the chemotherapy generate cross-resistance? So if you're treated with a chemotherapy drug, do the bacteria respond in such a way that if you then expose them to antibiotics, they're already pre-resistant in some way? And the drug I'm going to talk to you about today is 5-fluorouracil, a chemotherapy drug. It's a pyrimidine analog inhibiting DNA synthesis. That's its mechanism of action. And it's used intravenously for some cancers, but also topically, particularly for skin cancers. And we've been looking at this drug with respect to Staphylococcus aureus. Now, how does 5-fluorouracil work in this way? So a postdoc of mine put together this figure which shows how 5-fluorouracil is metabolized. And if we have 5-fluorouracil up here, so this shows the pathway through which it can be metabolized to other molecules. And the labels on the step here, where it says SA, that is the Staph aureus enzyme that catalyzes that reaction. And the H is the human enzyme that catalyzes that reaction. So you can see that you get the 5-fluorouracil is metabolized down through to inhibit this step here with thymidylate synthase, and that's going to inhibit ultimately DNA synthesis. But you'll notice this is also the same pathway that sulfamethoxazole and trimethoprim act on. So just from putting together the biochemical pathways, we can say, well, it looks like there's potential for an interaction here. But the question is there if we actually test that? So we just did some simple growth curves, essentially, using sulfamethoxazole, in this case I'm showing here, and 5-fluorouracil, and then a mixture of the two. And what was interesting, this is two different Staph aureus strains that are being shown here in these two different figures. The sulfamethoxazole by itself doesn't really inhibit the staph. It doesn't really have anti-staph activity, so that's not a surprise by itself. And the 5-fluorouracil is quite toxic to the staph. They don't grow, both of these strains. But if you mix the two drugs together, the 5-fluorouracil doesn't seem to be having nearly as much activity as it did. So there's an antagonistic interaction happening here. We don't know what it is yet. We don't understand the mechanism behind it, but there's definitely antagonism. So the interaction of these is occurring. We wondered if efflux might be involved in this as well in terms of the reaction. One of the generic ways that the bugs might be able to evolve resistance is just by switching on efflux. And this figure essentially shows that no efflux is not important. So this is your negative control here. So this is an assay looking at a fluorescent dye being effluxed out of the cell. So this decreases slightly because it just leaks out a little bit. This is a positive control. So this, if you switch on the efflux system, then the dye is pumped out very quickly. And then this is showing what's happening just naturally. And you can see all of the lines here are together, so it doesn't matter whether we've got the drug present or not. It makes no difference. So efflux is not involved in this process. So we wanted to do some experimental evolution, because I'm interested in evolution. It's an area that particularly intrigues me. And we did an experiment where we did 15 passages, and you get about 10 to 15 generations of the staph per passage. So we're looking at maybe about 150 generations of the bacteria at two different concentrations of 5-fluorouracil and one with no 5-fluorouracil, 4 replicates of each. And interestingly, we get a change in the sensitivity to the 5-fluorouracil in these strains. This is disk sensitivity data, so this is disk zones. Diameters, but we saw no difference at all in any of the antibiotics. So that seems like good news, but we're cautious because these endpoint assays are not very sensitive to what is actually going on in the organisms over time, and selection can act on very small differences. So that might not be the whole picture. We sequenced these strains as well, that we did the experimental evolution on, and this is where the genomics bit comes in. So we integrate genomics into these types of experiments. And what the bit to point out to you here is that where it says 6.3, 6.2, 6.4, and 6.1 on the far right-hand side there, that's our 4 different replicates in the experiment. Every replicate had a mutation in the same gene, but you'll see just by looking a bit to the left, they're all different mutations. So there is some kind of parallel evolution going on in these experiments, which means that disrupting that site seems to be important for their response to being exposed to this drug. And that makes sense. If we go back to our figure here, the enzyme that I just said we had mutations in is the one circled there. So if we are mutating that enzyme and we can't get through that pathway there, then we're no longer going to get inhibition of the system. And we're currently working to use some of the other intermediates in this pathway just to confirm that that is what is happening. So that was all very good and interesting, but what I was particularly wanted to know was about what happens if you've already got a resistant strain and you're exposed to the drug. So what we did here was we took 3 clinical strains of Staph aureus that are trimethoprim resistant. As far as we know, to our knowledge, they've never been exposed to 5-fluorouracil before. And we mix them 50-50 mixture with a sensitive strain, a sensitive lab strain. And then you expose some to a drug and controls that you don't expose to a drug, and you just measure the frequency of the 2 different types you get out at the end, so a simple competition assay. And what should happen is if you have no change in the frequencies, then you end up with a frequency of 1. So that's shown on the figure here. Oh, my pointer's disappeared. So where it shows 1 on the figure, the relative fitness of your your resistant strain, that means there's no change at all. If the mutant fitness goes below 1, that means that they're losing out to the sensitive strain. And if it is above 1, it means the resistant strain is doing better. And what we see is that if we have no treatment, which means no 5-fluorouracil, then the sensitive strains are doing slightly better. Now, that fits with the common hypothesis that if you have a resistance mutation or you're resistant to drugs, there's a fitness cost associated with that. We don't know that's definitely what's happening here, but nevertheless, the resistant strains don't do so well when there's no drug around. But as soon as we put 5-fluorouracil in the mixture, these resistant clinical isolates survive much better and they outcompete the sensitive strains that are there. So this is quite concerning because it means that use of the chemotherapy drug or exposure to the chemotherapy drug selects for existing antibiotic-resistant organisms. The other thing we noticed was to do with small colony variants. I'm sure many of you have heard about small colony variants, particularly from Staph aureus. They're a common phenomenon. And small colony variants are known for being more drug resistant and for being more virulent. And we noticed that on our plates, we would, for some of our strains, we would get a mixture of small colony variants and normal strains, shown on the left there, normal colonies. But when we exposed to 5-fluorouracil, we were only getting these small colony variants. So we wanted to know whether there was any difference with these small colony variants to the other strains. And so we did some MICs on those. So we've got small colony variants, which is the SCV, and normal colony variants, the NCV. And we tested against a number of different drugs, and we find that the small colony variants do have slightly raised antibiotic MICs, which fits with what we know about these types of strains. And we also see that if we add thymidine to these, that you can increase the level of resistance to these drugs. And that makes sense if we go back to this figure again. If we've added thymidine, then we've bypassed this step that might be inhibited, which means that the drug is no longer going to be impacted. So that all kind of fits together. So that's where we are at the moment with this piece of work. We've been able to show that antibiotic drugs and chemotherapy drugs, they can interact with one another and they can interact in the same pathways. We want to do some more work to work out exactly how that is happening. We've been able to show that chemotherapy drugs can select for pre-existing resistant strains, so that's particularly worrying and something we need to look at more broadly, and how wide an effect is this? Do you see it with other drugs? Do you see it with other species? Do you see it with other types of resistance? And we see that we can select for small colony variants. Those small colony variants that are more resistant to treatment, that are more able to survive, that could be a concern for what's happening in the treatment of these patients. So that's some of the work we're doing on non-antibiotic drugs and how they can be helping drive antibiotic resistance. I'm now going to move on to 2D and 3D bacterial chromosome structure and how that's related to drug resistance. So I'm going to go back to my favourite organism, Acinetobacter baumannii. Many of you will know this organism. It's a serious gram-negative infection. An opportunistic pathogen, so we tend to only find it in hospitals and then only in the sickest patients. It's not that common relative to some of the other organisms, but the reason that it's on the WHO bacterial priority list is because of its high levels of drug resistance. So that if you do get an infection with it, the chances of you surviving that are that much lower than with many other organisms, because we just don't have the drugs to treat them with. And that's why, particularly in low and middle income countries, we can have really high rates of mortality associated with this organism. So it's one that we really want to know more about the biology of and find out more about how it's becoming resistant, how it's adapting, and hopefully how we can then come up with new strategies to treat it. So I'm now going to go a bit beyond the way we normally think about bacterial chromosomes and the type of work we do with epidemiology and genomics. When we're doing our studies, we often will sequence the genomes of what we're looking at, and we identify whether certain genes are present or not. We might call SNPs in our strains to see what's changed in them. We might look at the mobile elements, maybe, and perhaps a kilobase or two size bit of DNA, and look at how those are moving around. But we don't normally go much further than that. That's kind of where we stop with all these kind of studies. And all those papers I showed you at the start that I published, they're all of that nature. They stop there. But really, I'm going to argue that I think we should think more broadly than this and think more about the biology of what's going on with these organisms. And I'm going to start by thinking about 2D chromosome structure, and then in a minute I'll come on to the 3D chromosome structure. So thinking about two-dimensional chromosome structure, we typically think of our circle, our bacterial circular chromosome. Most of them are circular. It's not all of them, but most of them are circular. And they have their origin of replication up at the top there, and then their terminus down near the bottom, usually roughly opposite each other, not always exactly, but usually pretty roughly opposite. And you can see the diagram on the left there. When you have a slow growth rate, you've got your replication fork coming down, and when you have a high growth rate, you'll have more replication forks. So the bacteria will begin a new replication fork before they've finished the old one, so you can have multiple replication forks simultaneously in the one cell. But what that means is, if you're a gene like gene A there, the one in yellow, that's near the origin of replication, you're going to have more copies of yourself in a cell as the cell replicates. And if the faster it's replicating, even more copies. If you're gene B, about halfway down, you'll have a sort of intermediate number of copies. And if you're gene C, the green one down near the bottom, you're going to have the fewest copies. So where you are, as if you're a gene, where you are in the chromosome affects how many copies of yourself there might be in a cell at any given time. And we know that copy number, or gene dosage, as it's called, affects transcription levels. So if this was antibiotic resistance or virulence, then that could be really important for how these organisms respond. And this is all dynamic. It changes as the growth rate changes, which is impacted by all kinds of different things. We also know that bacteria are very good at rearranging their chromosomes. So they do lots of different types of rearrangement. We often look at small arrangements like the movement of transposons or insertion sequences, that kind of thing. But if we think more broadly, we can categorize rearrangements into a couple of categories, which we call balanced rearrangements. So these are things where the total content doesn't change, but things have moved around. So that would be things like inversions and translocations. Or unbalanced rearrangements, so where the content has changed in some way. So that's where you've got your insertions and your deletions and your duplications. The most common of these by far, if you look in the literature in this area, are called replication-associated structural rearrangements. And these happen around the origin of replication and the terminus, and it's very common that large fragments, tens of kb in size, just flip orientation around either the origin or the terminus. We don't know why, and we don't know the impact, but we know if people have studied this across a huge number of different bacterial species, and it seems very, very common. They do it a lot. So we get these large-scale rearrangements. And we wanted to know what kind of impact might these rearrangements be having. So I've got a PhD student who's currently in his first year. In the UK, with PhDs are 3 to 4 years, so he's just fairly at the start of his project. And he's going to be looking at this in Acinetobacter baumannii. I'm going to present a bit of what he's done so far. So the first thing that we did, I say we, I mean him, really, Matthew. We are only using long-read sequenced genomes for this because we need to know that these structural changes are actual biological changes and not to do with some kind of assembly error or something like that using short reads. So we're only using long-read sequencing. And we've taken from the database 494 long-read complete genomes, so they're single contig genomes. And first thing we did is just phylogeny, because I like phylogenies, and because we wanted to just check that we get the population structure we would expect for this species, which we do. You can see from the figure here, the large orange clade around the sort of bottom and side, that's international clone 2, which is by far the most common clone in this species. And then we have a number of other clones represented by the different colors. And then we have some sequence types which we've picked out which aren't associated, most of them, with a particular international clone. They potentially represent new international clones. We're beginning to look into that. But nevertheless, with this data set, with this approach, we can get a good overview of the species. So we think our data set's reasonably representative of the species. The first thing we then did is look at rearrangements around ribosomal RNA operons. So bacteria have multiple copies of their ribosomal RNA operons. In Acinetobacter baumannii, they have 6 copies. But because those are identical copies, then they're sites at which recombination can easily happen, because you've got that same bit of identical sequence, and those are the sites where recombination can easily happen around. And so the first type of analysis is looking at, okay, if we just break our genome down so that each of those lines there is the whole genome, and these are drawn to scale, you can split them into fragments based upon where the ribosomal RNA operons are. So the gaps in between each arrow is where there's a ribosomal RNA operon. And then they're just numbered, and then you can look at how they are in different orders or different locations in your data set. And these are the different structures that we find in that data set that I just showed you in the tree. We have a number of different arrangements that show either a change in orientation or a change in position of large fragments of the genome. So that's good. So we can see that, yes, in our data set we do have variation in this 2-dimensional chromosome structure. However, this is quite large scale, quite zoomed out, because we're only looking at changes that happen around these ribosomal RNA operons. And bacteria, of course, are going to exchange lots of other bits of material in other places. So how can we look at that? So what Matthew has done, and don't ask me the technical details of the maths of this because I don't understand it, you'll need to speak to him, but I'll try and explain the principles to you. What we've done is convert the genome into integers, so integerization. And that's the cartoon at the top there is showing this. So what the analysis does is it looks at all of the genomes and it looks for conserved blocks of sequence across all those genomes. And a conserved block of sequence, it then just delineates and it gives it a number. So in the example at the top there, you can see these 2 genomes. It's found 3 blocks of conserved sequence, and it's just given them a number. So the top one arbitrarily is 1, 2, and 3, which means that the next one is 1, 3, and 2. and they're all say plus. If it said a minus after the number, that would mean that the block had switched orientation. So we get a number code for every strain of a long string of numbers, either plus or minus, which tells us what the structure in terms of fragment order there is in those genomes. And then you can plot it like this. And so what this is showing is the total occurrence of each of these blocks. Each of those blocks is on the x-axis. And the color code is by the number of copies. So most of them the blocks are only in one copy. And then there's lots that are in no copies. So there's only a few strains that have that block. So you can think of that like we would think when we do our normal genome analysis, we think about a core genome and an accessory genome. So that bit on the right-hand side, the big orange block, one copy of each of those blocks, you could think of that as like a core genome. And then the other stuff, where there's lots of blue, there are not many copies of them, that might be more accessory things that aren't found in many strains. And they're ordered along the bottom there in terms of what's called parallelism, from high to low, which means low parallelism means that they are found grouped together within the phylogeny. So that's what you'd expect from core genomic type areas. And then high parallelism is stuff that you would find in different areas of your phylogeny, so that might have moved around. So that's lots of data. What does it mean? What can we infer further from that? So what Matthew has then done is he's looked at calculating rearrangement distances. Once you've got this information, you can then work out, if you think of it a bit like a jigsaw, how many pieces of the jigsaw do you have to move to get from one picture to the other picture? And that's your rearrangement distance. And then you can plot a heat map, a bit like that. So sorry, the dendrogram on the side there hasn't come out very well. What this is showing is those rearrangement distances. And plotted on the top up here and on the side are the same coloured clades that we looked at in our tree at the start. So the large orange block is international clone 2, for example. And the other coloured blocks are the other international clones. And the darker the colour, then the shorter the rearrangement distance is, so the less bits you have to move to get from one structure to another. So we're talking about the structures here, not the sequence. So as we would expect, which is good because it means this method is replicating what we would expect to see biologically, you get far fewer, so a darker colour, far fewer rearrangements happening within a clone than there is between the different clones. But there are some examples, like you see in the large orange block at the side there of international clone 2, there's some grey bits. So these are strains that are not part of global clone 2, but they have the same genome structure as global clone 2. So the content is not the same, but the structure, the way they order it, is the same. So this is what we want to get at, about whether there are particular structures that are particularly advantageous, that can help the bacteria either survive the stress or cause infection in different sites. We wanted to think about this in terms of the different clones and also the different sites of infection. So this just summarizes, if you take that information and summarize those distances, average them, and you look across the different international clones, you can see that international clone 2 here, the second from the left, has relatively low distances in between the different members of that clone. Some of the other international clones have got quite high distances between them. Now, we don't know if that's a function of some of those clones being younger and perhaps are more diverse and haven't been selected, bottlenecked down to a more particular structure, or whether it's some other biological function. And that's something we need to start looking at more now. We also looked at the site that these came from, because our hope was that we would find, if you have got something coming from bloodstream infection, for example, there's a particular genomic arrangement that's good for infecting the blood. Maybe there's another that's particularly good for infecting or for surviving in the water system or something like that. We can see differences in this, and if you do statistics on it, you get statistically significant differences between them. I'm skeptical as to whether that means anything, for two reasons. Firstly, because some of these groups are quite small in terms of the number of isolates involved, and so that could just be skewing things. And secondly, this relies on the metadata that other people have inputted into the database. And relying on other people's metadata is notoriously a bad thing to do. You don't know what someone means when they say, if you take the water category, for example, there are 66 in that water category. Well, water can mean from a pristine lake high up in the mountains through to the hospital water pipe that's coming out with all the effluent in it. They're both water. So it's not really a fair comparison. There's no way of us knowing from the database what people actually meant. So I think there's more to look into there about what effects there might be going on. So what's Matthew doing next? Well, we have a collection of historical isolates that I actually inherited from our PhD supervisor when he retired. And with collaborators, we have long read sequenced those. And Matthew's, I asked him by email yesterday how many we had, that he's just finished doing the quality control of those genomes. So there'll be 226 of those that we'll add to this collection. And they've been collected from about the 1980s through to the early mid-2000s. So they should allow us to have a much better look at the longer term evolution of this species. And then we're going to get all the other complete genomes we can from the public database. So that's, so I said before we were only using complete genomes. Those were long read sequence genomes put before. We're now going to add to it all the other genomes, regardless of sequencing technology, that are considered complete. And our estimate is that's about another 2,200 that we'll add to this data set. So I guess that takes us up to, what, just over 3,000 genomes or something like that, will be the final data set that he's then going to take forward for his analysis. He'll have to repeat what he's already done with the larger data set, and then we'll move on and look at the detail of which genes are actually involved in these fragments that we've seen moving around, which genes are on those fragments, which genes are moving the furthest distance. We should be able to calculate some kind of metric that shows which genes are moving the furthest, and whether that's towards the origin, where you tend to get more highly expressed genes, or towards the terminus. And therefore we should be able to infer an effect on gene expression. And this won't happen in Matthew's PhD, but then perhaps a future PhD student can actually then go into the lab and we can measure gene expression and see how that matches up with our predictions. And then the other thing we want to do is large-scale population genomics analysis. So we want to look at selection, which genes seem to be under positive selection or purifying selection, and see if we can work out what's happening between these different clonal lineages within the species. So that's two-dimensional chromosome structure. But what about three-dimensional chromosome structure? So the bacterial chromosomes, we in our textbook, we have a nice circle with our origin and our terminus. That's not what they're like in real life, but they're also not just randomly tangled up inside the cell. They're highly structured and organized. A whole load of different proteins, different types of DNA binding proteins that hold the chromosome in a certain structural orientation. And we know that depending upon which part, how that structure is put together, that affects which genes can be expressed and how efficiently. So this three-dimensional structure is really key to gene expression and just how the organisms survive and live on a day-to-day basis. So it might not just be important where you are, if you think of the chromosome as a circle, where you are in that structure, but also once that's then folded up, you could be a gene that is perhaps near the terminus, but if you're folded up, you might be near a gene that's near the origin in physical space. And does that matter? We can start to look at that now. People have been doing this for a little while in eukaryotes. Only more recently have people started doing this in prokaryotes. And we use something called a chromosome contact map. And this allows us to actually measure which bits of the DNA are more close to each other in physical space than others. And how this works, essentially, is that it's through a sequencing technology And you take your bacterial cells, and the first thing you do is you add a cross-linker, which binds all those proteins to the DNA and holds them firm in place. Once you've cross-linked your DNA, you then just digest it with standard digestion enzymes. Then you dilute it down so that, in theory, when you then add in a ligase, only the bits of DNA that a protein is holding together, only those bits of DNA should be close enough to each other to bind back together. So what you get is a bit of sequence that's a hybrid of two bits of DNA that were near each other in physical space. You can then put all of that through a sequencer, sequence the whole thing, and then through some clever bioinformatics tools that, again, I don't know exactly how they work, but they're able to identify that point in the middle where you've got that hybrid. They can separate it out, you can map back to your genome, and you're then able to draw a contact map, which is a bit like a heat map, to identify what they called SIDS, chromosome interacting domains. And there's a cartoon here. This shows what it looks like up on the right there. The bit at the bottom is a cartoon of what's happening with the chromosomes. So you've got the two of these loops, and then another loop that's a little bit further away. And you can see that above that, in the contact map, the bits representing the loops are the darker coloured triangles. And then you can see, because two loops on the left are a bit closer to each other than the one on the right, there's a slightly larger, slightly darker triangle that links those two. So the software is able to look at that pattern and predict that structure from these patterns. So we've done some initial work on this. This was a student who joined me from a lab in India. So she was doing her PhD project in Delhi, in India, and she had one of her programs where she had 3-month placement somewhere else in the world. And so she came to us and did this. She was interested in adaptation to the antiseptic triclosan in E. coli. And she'd done a whole load of experimental evolution to look at this, and then we wanted to look at the 3-dimensional chromosome structure. So what we're seeing here is, on the left is the wild type organism, and on the right is the one that's been adapted to triclosan. And I always find, looking at these in person, it's hard to see what's going on, but the programs are able to pick out, through their statistical inference, these different interacting domains. And it's highlighted in yellow, these different SIDS, these different interacting domains. And you can see that there's a big difference between the ones just in the wild type with no drug exposure that they haven't been adapted to, and the one on the right that's been adapted to the triclosan. So they've changed that 3-dimensional structure. And we've highlighted there the FAB-I gene because that's the gene that's responsible for resistance to triclosan. And it's found there right on the edge of one of these chromosome interacting domains, which fits perfectly with what we would like to have seen, because it's known that the genes at the boundaries of these interacting domains are highly expressed. So that makes perfect sense in terms of the response to the drug, that they've now changed that 3-dimensional structure, so the gene that they need to highly express to get resistance is on the boundary of one of these areas. Not only that, but when we look at the mutations that happened in these strains, we've got a couple of different types of mutations here. We've got the SNPs in the top bit of the table, and then structural variations, insertions, and duplications. You see that in all cases, they were found within one of these SIDS, and that's shown on the figure on the right there. That's the chromosome, and we've got the duplications in purple on the outside. The insertion is a little red arrow, and the little I above the blue B box, that's the SNP. All of them were found in one of these chromosome interacting domains. So all of the changes happened in those domains, and therefore that 3D chromosome structure seems to be linked intimately with where they're changing their structure, where these mutations are happening. So that leaves us with a whole load of open questions, far more than we're going to get a chance to look at. But where we're going to start is thinking about this more with antibiotic resistance. So how do these 2D and 3D structures change under that antibiotic stress? trying to really characterize that. How does the movement of AMR genes, that we all know that they do, how does that alter their expression, and how can it alter their gene dosage in the cell? How does movement of the AMR genes alter their physical proximity within the genome, and whether they're linked with more highly expressed genes? Something we know is that when you look at particular functions of the cell that are important, in terms of the three-dimensional chromosome structure, it will cluster together genes that are involved in the same biochemical processes, so that all the machinery to do with high levels of transcription translation are physically located in the cell. They don't have to move to different parts of the cell to express different genes involved in the same pathway. So is that linked? Is there something similar with AMR? And given all of this, and given the importance it seems to have, does that mean we can identify new targets, new ways that we can think about disrupting some of these cellular processes that's specific to bacteria that we can use therapeutically? So that brings me to the end of what I wanted to show you today. I've just got a few acknowledgements. Firstly, the people who did this work. It's always the people in the lab who do the work rather than us professors sitting in our offices. So thank you very much to them for generating all of this data and all of their hard work. Matthew, Sarah and Layla. I want to thank my collaborator Gemma. She is in the Quadram Institute just next door to the building I'm in. She's also working in very similar areas to me. She's looking at it in salmonella. Dipanata is the visiting PhD student from Delhi, who I mentioned. I want to thank my funders, who are the Wellcome Trust and the University of East Anglia, who funded the work I presented to you today. And I particularly wanted to thank Andres for inviting me here, to be here to talk to you, and also to the British Embassy, who funded the trip so that I could be here. So thank you very much for your attention, and I'm very happy to take any questions.
Great. Thank you very much, Ben, for this extraordinary talk. It's very interesting. Many things to explore and to dig in. So we are open now to some questions of the audience. If someone wants to do some question or comments, I can start with some, just to break the ice. In the case of that you shown at the very beginning, you shown a little pot of, or little tube of bleomycin.
Yes.
Where are your thoughts about bleomycin and resistance to carbapenems mediated by NDM? As normally these genetic elements, carborine NDM, are associated with bleomycin resistance.
Yeah. So I had a PhD student who worked on this very topic. For those of you who aren't familiar with the NDM transposons, NDM is a beta-lactamase, a carbapenemase, really important in multi-drug resistant gram-negative organisms, causing a huge amount of problems worldwide. Immediately downstream of that gene in the transposon that carries it is a gene that's annotated as a bleomycin resistance gene. It's always there. It's always the same as well. It doesn't seem to mutate. It doesn't seem to vary, which made me think, well, it must be important for something then, right? Because if it wasn't important, you'd expect to see some variation in it, some SNPs accumulate. Maybe every now and then you'd see some with a deletion or a bit of it being chopped off. But it's always there. So we did this. We tried to make constructs which had, so fluorescently labeled constructs, which had just the NDM, just the bleomycin, or the pair of them together, and marked them with red and green, and with the idea that we then do competition experiments, put them through a flow cytometer, and be able to detect the changes in the frequencies. COVID happened, and we had to rush the very end of her PhD, so we haven't really got to the bottom of that. We haven't been able to properly do those experiments with those constructs. We haven't properly checked that they are that they're structured as they should be and working okay, that we can then do the experiments. But it's absolutely something we want to look at, because it's not if you start looking, there are quite a few other plasmids are mobile elements that have a gene that's annotated as a bleomycin resistance gene.
Yep.
Now, just because it's annotated as that doesn't mean that's what it does. That's always something to bear in mind. Don't believe annotations always, but that's what they're annotated as. So yeah, they do seem to crop up. There's one in a plasmid which I bunged into. I did a quick dirty experiment just to see what might happen, and I bunged a plasmid. I think it was from a bacillus that has a bleomycin resistance gene just into another one, and it immediately just grew. The strain just grew better, which is the opposite you expect. Normally, if you put a plasmid in something, it should grow worse. It immediately grew better. So I think there might be something there.
Yes.
But yeah, we haven't gotten to the bottom of it yet with the bleomycin.
Nice. Thank you. So also, if you want to do a question, to make a question, so don't worry if it's in Spanish, I can translate it. And also there is a question from the audience online. I just got this. We are so sophisticated. So there's a question saying that, well, it says that, brilliant talk. I was just wondering if you know for how long bacteria keep their antibiotic resistance once they are not longer exposed to antibiotic drugs.
Sorry, you said
For how long bacteria keep their antibiotic resistance once they are not longer exposed to antibiotics?
how long they keep
Yes, they keep the resistance level after you remove the antibiotic or are not exposed anymore to antibiotics.
That's very variable on a case-by-case basis, I think. So there, in many cases, you take away the selective pressure, and then your resistant strain can be outcompeted by the sensitive strains. But then in some instances, you'll get compensatory mutations happening, which means that then the fitness cost of that resistance is decreased, and that strain is no longer outcompeted by your sensitive strains. And that's why over time we see the multi-drug resistant strains we do, because they've adapted so that the fitness cost is low enough that they're able to survive. That's an interesting way of, or an avenue of investigation, is trying to look at how we can come up with approaches that perhaps don't target things that have a fitness cost so much, so that they can't evolve a resistance to it. Antivirulence approaches are one of the strategies that people have been looking at, because the idea is that if you've got something that affects virulence, then the obvious thing for the organisms to do is to evolve to become less virulent. So either way, we win, right? That's the idea. So there are ways of looking at it to try and make those things play in our favor.
But also maybe consider other factors, like, for example, if you remove antibiotics, but the resistant genes was in the same class with genes mediating resistant to quaternary ammonium compounds or heavy metal, maybe they will persist.
There's an awful lot of co-selection. Yeah, you only need, I mean, the experiment we did there with the 5-fluorouracil, 5-fluorouracil is selecting for the trimethoprim resistant strain. As far as we know, they don't have any 5-fluorouracil specific resistance genes. That kind of co-selection, you have a resistance plasmid and it's got 5, 6, 7, 10 resistance genes on it. You're going to select all of them with just a single drug. So there's an awful lot of that going on. Yeah.
Great. Any questions or comments? Max, you will have to for the streaming. No, that's okay.
Respecto al experimento con 5-fluoracilo, si es que las concentraciones en las cuales encontraron selección son similares a las que se alcanza con el medicamento aplicado en una persona.
Okay, so the question is related with your experiment with these 5-fluorouracil compounds. If the concentrations that you used in vitro are quite close to the one that are reached in the plasmatic level in the organism.
It's funny, I can't speak Spanish, but I was anticipating that question. I could tell that's what you were asking. Yes, so the concentrations we use we use two different concentrations. One is the concentration that you might find, or is similar to the concentration you might find in the blood. The other is, so many of these drugs you can, if you take certain tissues, they accumulate in certain tissues, so you can get much higher concentration. So that depending on how you look at it, there's a big concentration range. So the higher concentration sits sort of roughly around that sort of concentration that you can get of accumulation in certain tissues. What we haven't done, and I should say that that's a really important point, and it's something we're very keen on doing, is making sure we're using physiologically relevant concentrations of these drugs, because many, particularly these older papers that have looked at these, have used really high concentrations, and then yes, you show an effect, but so what? You know, that no patient ever sees that concentration of drug, so does it matter? So we do try and use physiologically relevant concentrations. Having said that, we know from a lot of studies on antibiotics that you can have really very low concentrations of antibiotic, can still select for resistance and select for resistant strains. It's concentrations far below the MIC. You only need a very small difference in fitness to be able to start to see a change in that and to see that selection. And that's something we want to then do more of now. From the point we've got now, we can then do more longer term experiments with lower concentrations and see over a longer period of time what kind of selection do you get happening, because these were only run over, what did I say, 6 passages, or no, 15 passages, wasn't it? So, you know, just a couple of weeks, which isn't that long really. And we all did it on liquid culture, and so you can say, well, how relevant is that to you know, real life? So we have a study. I've got a master's student at the moment who is looking at Staph aureus we've isolated from actinic keratitis. So they're the lesions you get on your skin before it becomes the sort of precancerous step. And we're trying to see whether we've tried to get samples from patients before and after they've been treated with 5-fluorouracil cream on them, and we're going to put those into a cell model and see if they are different in any way in terms of their virulence. We can test them in terms of their antibiotic sensitivity. We can sequence them and see if we can see any differences from a sort of more real life clinical point of view. But that's a very small project, so that's something that again we'd want to expand upon.
Nice. Yep.
Yeah, about the 2D analysis structure. If with that analysis you can trace back like gene flow between genes, for example, a gene flow from a population to another, something like that.
So we can work out which genes are on which fragment. We should be able to then look at gene flow. We'll need to, once we've got our complete data set and our complete phylogeny, we'll need to then look at see how good our dating information is and our metadata that allows us to, we'd like to construct a dated phylogeny and look at that. But then, yes, we should be able to then look at seeing the gene flow between the different clades within the species, or even just different individual strains if there's some particularly interesting element that's moving around or fragment that's moving around. We should be able to trace that and see what's been happening, both trace that over time, which is what we ideally want to do. Yep.
Where I can use that, like distance, custom distance algorithm of blocks, where I can find that?
The software.
Yeah.
So I only remember one of them. One of them is called PariBrick. If you want to come to me afterwards or take my email address or whatever, send me a message and I can get Matthew to give you all the details.
Great. Great way to interact.
Exactly.
Another question? We have time for a couple of extra questions. Comments? Vicente.
So, thank you very much for the presentation. I want to ask you about when you analyze the DNA structure and when you do the crossing into DNA, are you looking for specific DNA binding proteins, or you have all of them and then do the
So it's all of them. So it will just indiscriminately, any DNA that is bound together with proteins will get fixed in place. Yeah.
and there is some that special?
Don't know. Don't know is the honest answer. There are a number of different classes of DNA binding proteins that do slightly different jobs. interestingly, one of them, HNS, seems to be found on plasmids sometimes. And there's a paper I noticed the other day that I need to sit down and read that started looking at that, because I think that's really interesting. We don't know whether any of these are more or less important in terms of the drug response. But that's something we could start to look at. We could make knockouts of the different DNA binding proteins, see how that affects things. There's a huge number of options and opportunities here for what we could look at, but that would be really interesting to see.
Because you can immunoprecipitate them.
Yeah. Yes, exactly. So you could do that type of approach and target the different types, and then see which areas that they're binding and whether they are the ones related to drug resistance or not. Yeah, absolutely. Yeah.
Great, thank you. And we have time for the last question, and after that we are going for the official picture of this activity just outside the building. So one more question or comment. No, I have a final one, if you, let me. The advantages of being the moderator. So, also, just not in terms of these cancer drugs or these drugs to treat cancer, but there are also some evidence about how the artificial sweeteners are also interacting in some way with antimicrobial resistance. What are your thoughts about that? What do you believe about that?
No, that's a really interesting area. I was a second supervisor for a student at Anglia University who was looking at artificial sweeteners and potentially how they were. She was looking more at how they impact the gut lumen and damaging that, but then my role was saying, well, they affect bacteria too, and so what's the interaction there? Yeah, I think that's another one of these categories of drugs that we use all over the place that have these properties, and actually we don't know what they might be doing, the details of it. I absolutely think we should be looking a lot more of that, doing some experiments of these type selection experiments, cross-resistance experiments, and competition experiments to see what sort of concentrations these different sweeteners might affect different species and different types of mechanisms of resistance, and therefore whether we should consider how we alter their use in our food.
Yes.
Yeah.
Yes.
Yeah.
Okay. So last chance. Always. Well, thank you again. Thank you very much for your
Thank you
presentation.
for your attention. Thank you.
And also, we do have a wee gift for you. Because in Scottish word. So thank you so much for your presentation. So here is your present. So I hope you will like it. So thank you again. We have now the official picture outside the building, and then we have a